Uh, so very good evening to all the students. So and my dear senior colleague, sir, Dr. Manor Dadwad sir. So today we will learn about the fungal diseases of vegetables and their management. So for first hour, we'll see these fungal diseases of vegetables, and we'll take a break for 5 to 10 minutes, and we'll rejoin, and we'll continue for the viral diseases or that viral lectures on architecture, composition, and classification of plant viruses. Okay, so yes, we know all that there are n number of vegetables grown in India, uh, like potato, tomato, chili, radish, carrot, turmeric, onion, peas, ginger, brinjal. So these are the different type of vegetables which are grown in India. So we will see, for example, cucumber. These are the scientific name of these different vegetables: cucumber, Cucumis sativus; bitter gourd, that is Memordica charentia; ladyfinger, that is bendi; Abelomoscus esculentus; radish, Raphanus sativus; ridge gourd, Lufa acutangula; then red spinac, Bacilla alba; spinac, Spinacia oleracea; carrot, Decus carota; then American cucumber, Cucumis melo; then pumpkin, Cucurbita maxima; bottle gourd, Legionnaria, Ciceria; then Edo, Colocasia esculenta; cabbage, Brassica oleracea; pigweed, brinjal, cauliflower, and tomato. So and so forth. So these are the different vegetables which are cultivated in India, and these vegetables faces many biotic constraints, that is diseases. So the fungal diseases plays an important role in the uh, reduction of yield and causing heavy losses in different vegetables. So we'll see the major fungal diseases of different crops. We'll start with the potato. Potato is one of the most destructive diseases. Lead blight of potato is one of the most destructive diseases of potato, and which was recorded in the early 19th century. So in the Andes Mountains in the South America. So this is one of the economically important diseases which has caused huge losses worldwide and in India in particular. So this fungus initially moved from South America to the Europe during 1842, and it established in Ireland, where it caused the epidemic. That is, we are known about this. The infamous Irish famine is caused by the lead blight of by the Phytophthora infestans, causing the lead blight disease of potato. So this disease has, and since then the disease has studied, and during this infamous Irish famine, so more than 4 million people were directly affected by the this pandemic. So now we are aware about the terms like epidemic, pandemic. So okay, so the disease which is having the worldwide distribution, which are called as a pandemic diseases. The diseases which are called in a particular country or region, they are epidemic. So like coronavirus, now we are calling it as a pandemic disease. So these are the diseases called based on their area of occurrence, and how much area or region or continents or states they are infecting or affecting the people or plants. So that's why these terms are used. So the systematic growth of the plant pathology as a discipline was started after the epidemic of the disease. Excuse me for a while. Okay. The disease has worldwide occurrence. In India, the disease was first reported in 1980 from Darjeeling district of West Bengal, and by 1900, that caused epidemic in Indo-Gangetic plains in 1913. So, in India, the disease has caused loss of around 10 to 75% in different parts of the country wherever potato is cultivated. So we can see the symptoms of the late blight disease of potato. So the first, initially, the symptoms of late blight in the field are small light to dark green, circular to irregular shaped water-soaked lesions or spots appear on the leaves of the potato plants. These lesions are usually first appear on the lower surfaces of the leaves. Then these develop near the leaf tips or edges where the moisture is there. So for a long time, so there the pathogen starts multiplication and starts showing symptoms. And these pathogen you can see in the picture that, so this is the spots which is caused by the Phytophthora infestans, and the spots is surrounded by slightly yellowish zone around the fungal spores, the fungal spots. So in and these are the different you can see in the late blight infected plants, which shows the symptoms starting from the lip tips. The lip tips, then lip tips, these spots foliates join together and they form the larger necrotic lesions. Larger necrotic lesions turn cover the entire leaves, and these initially these are like water soaked lesions. So and these leaves get necrosis, and starting this spread of this pathogen to the stem and other plant parts. So after the severe attack of the pathogen, the entire plants get destroyed, it get dried, and then after the infection is also visible in the potato. So in infected potato, you can, if you split the infected potato into two halves, it shows that there are some discoloration, uneven type of discoloration can be seen in the potato tubers. So on the surface of the potatoes, these slightly depressed areas that can vary from brown to purplish, or which are variable in size, which are visible on the skin of the potato. So these are the different type of symptoms which appear in the late blight infected potato plants and tubers. So now coming to the pathogen, we know that Phytophthora infestans is oomycetes, which is causing the disease. And the mycelium of the oomycetes is that cynocytic hyaline. Hyaline means the mycelium which is uh, transparent. So we are calling it as a hyaline. It is a branched, having different branches. This structure you can see in the picture, that this structure is of the conidiophores. So conidiophores, these are the we can say sporangiophores. These are the structures of sporangiophores. So these are the sporangiophores are also hyaline. These sporangiophores, on these sporangiophores, these sporangia are produced. So someone has raised a hand. Yes, anyone wish to talk? Okay.
Somebody has named himself as the father of plant pathology, and he has raised the hand. Please,
Yeah. Yes.
please unmute yourself and ask if you have questions. Some of you may continue. I think it has. You may
Okay.
have raised hand.
Okay. We'll go ahead. Later we'll discuss. Okay, these are the type of sporangiophores. These sporangiophores are hyaline, simple, they are branched. Septate means they are divided into different compartments, and these are the different type of septa we can see. So therefore it is called as a septate mycelium. And that sporangiophore is swollen, having swollen base. So it is swollen at the base and it is slender towards the tip. So this is the characteristics of the sporangiophores of the Phytophthora. Why I am telling all these, because there will be always a question, what is the difference between the late blight of potato and early blight of potato. So that's why these are the distinguishing, or these are the key features to distinguish the Phytophthora infestans from the alternaria. So you can remember these the distinguishing characteristics of the Phytophthora infestans, that is late blight of potato and early blight of potato. I'll cover that in the later slides. So these are the different types. These are the different sporangia, or this is the singularly we can call it as a sporangium, and plural we are calling it as a sporangia. These are the lemon shaped sporangia which are present in the Phytophthora infestans. They are thin walled and papillate. Papillates means these are having some beak like structures at the top, so that's why they are called as a papillate. Or this sporangium is also hyaline, it is transparent. Now we can see the disease cycle of the late blight as or the spores get overwintering, and the infected tubers, the mycelium from the tuber infects to the seedling. These are the sporangium, these are the sporangiophores on the infected seedling, and these sporangiophore is bearing the sporangia. So these are the sporangia. These sporangia get dispersed from the infected plant parts by wind or by splashes of rain, and they get dispersed from they get detached from the sporangiophore, which is come across, which has emerged from the stomata, leaf stomata, and this is the sporangium which is having the beak. So it is a papillate sporangium, and these sporangium, there will be two ways of germination of the sporangium based on the environmental conditions which are suitable or which are not suitable. So when there are the conditions which are suitable for the germination of the sporangium, these sporangium germinate and they directly form the germ tube and infect the leaf tissues. Whereas there are certain conditions which are not favorable for the sporangium, then these sporangium get it will, when there are certain conditions which will be favorable for after some time, then it will get it will mature and it will form the zoospores. Zoospores are the motile spores which are present in the sporangium. So they get the sporangium get burst and the zoospores get released, and these zoospores get transmitted to the wet leaf area where they can germinate and start the infection. So this one is the infected leaf. Again, this infected leaf is transmitting these from this infected leaf. Again, this sporangium get dispersed and they get this cycle get repeated. So in another cycle, that sexual stage of the cycle is zoospores are formed. The zoospores directly germinating on the they are transmitting from one plant to another plant. So these zoospores germinate and they cause the infection. So in case of the sexual reproduction, there are two types of mating population is required, that is oogonium and anthridium. Oogonium is the female structure and anthridium is the male structure. So they undergo the meiosis, that is the process of cell division, and they be followed by karyogamy, that is the exchange of nucleic material from one parent to the another parent, and they form the oospores. So oospores are the circular spores. So these oospores are the called as a resting spores, or they under suitable environmental conditions they get germinate and they form the sporangium. Again, the sporangium is causing the following the two pathways as I said earlier. So for management of the late blight disease, so we need to plant the disease-free seeds or tubers. So we need no need to mix the seed lots because cuttings can transmit the late blight disease. So use a seed piece fungicide treatment which is labeled for control of late blight. As I said yesterday, lecture that for application of any chemical in any crop, we need to follow the CIBRC released. So Central Insecticidal Board for registration and registration committee, which allow the application of chemicals in different crops by testing it nationally and internationally. So they allow use of any chemical in any crop. Okay, so we need to follow the CIBRC list. So blindly we cannot recommend to the growers, or we cannot say the management of diseases based on our presumption. So we need to follow the guidelines which has given by CIBRC. So we need to follow the list of chemicals which has a label claim in different crops. Okay, so use a seed piece. So avoid the planting. So that problem areas that may remain wet for extended periods or may be difficult to spray. So avoid the excessive irrigation time. There is some spelling mistake. So we need to avoid the excessive irrigation, because if there is an excessive irrigation, then the chances of infection of from one plant to another plant gets maximized, and the heavy losses can be incurred. So we need to eliminate the sources of inoculum. That is first is the primary source of inoculum, as a which is hairy nightshade weed species and volunteer potatoes. So it is grown on these volunteer potatoes or infected bulbs or infected tubers or infected plant part or debris. So we need to eliminate these sources of inoculum. So using of the we need to follow the foliar sprays of fungicides. We need to destroy the hotspots of the lead blight. So if any confined area is affected by the lead blight, so that part has to be completely destroyed, and crop rotation need to be followed, and all the cultural practices need to be followed, so that the pathogen, the non-host crops can be planted there, so that the pathogen cannot survive or multiply. So kill the vines completely 2 to 3 weeks before the harvest. Application of phosphorus acid to potatoes after harvest and before the peeling can prevent the infection of the lead blight in storage conditions. So we need to monitor the home garden and market tomatoes or potatoes near these tomatoes, as I'm saying why, because these lead blight is uh, so for the lead blight can move from the local sources of to potato fields. So these are the symptoms of potato blight or Phytophthora blight in tomato. So that's why we are managing the diseases together. So lead blight in potato and tomato and many other solanaceous crops are affected due to this disease, Phytophthora infestans. So these are the symptoms you can see in the tomato. So these are the different symptoms appearing on the, these are the water soaked lesions appearing on the tomato fruits starting from the early stages of their crop growth. This one is the water soaked lesion appearing on the stem. These are the in the green tomatoes. So this how devastating is the Phytophthora infestans in the tomato. So these this pathogen causing the severe losses in these 2 to 3 diseases, solanaceous crops. Okay, now we will see the early blight of potato. So early blight is another important disease of potato, which is caused by Alternaria solani. The disease has a common occurrence and it is known as early due to because its appearance occurs 3 to 4 weeks after the planting. It is prevalent throughout the potato planting and cause the huge losses. So in Germany and Denmark, it has reported to be causing more than 50% losses, while in Israel it was around 25%. The pathogen has a wide host range and causes infection on other different crops like tomato, chilies, brinjal, and many other solanaceous crops. So now you can see the symptoms of early blight of potato. So this is the distinguishing symptoms from the late blight of potato. Now you can differentiate the based why what is the difference between the early blight and late blight. So in symptom itself there is a difference. So starting from the pathogen is again a different pathogen, that is Alternaria solani. So and the symptoms which is caused by Alternaria solani are totally different from the Phytophthora infestans. So these Alternaria solani, which is causing the concentric rings on the leaves, you can see these are the concentric rings which are caused due to the Alternaria solani. These rings join are in severe condition. These rings join together. They cause the start. These are the necrotic tips. So these leaves, these spots join together, coalesce, and form the necrotic spots, yellowing of the leaves and complete necrosis of the leaves. And these it get spread to all the plant parts, and they get the foliage of the plants get affected due to this disease, because this disease is coming in the early season. So similarly, this disease is causing early blight in potato as well as in tomato. So you can see the similar symptoms are seen in the tomato plants. So these are the concentric rings, concentric rings appearing on the leaves, stem, fruits. So you can see the severity of the early blight in tomato. Okay. So now coming to the pathogen of early blight of potato or tomato, that is Alternaria solani. So you can see in the right side in the picture that these are the spores or the conidia of the Alternaria solani. So the mycelium of this fungus is septate and pale brown in color. Conidiophores, they arise singly or in small fascicles through stomata. The conidia are solitary, obclavate, oblong to ellipsoid, and tapering to a beak, muriform, pale to brown in color, having transverse 9 to 11 septa and few longitudinal septa. You can see these. This one, the conidia of the Alternaria is the septate, because these are the different septa. You can see the 9 to 11 number of septa are present. 9 to 11 transverse septa. These are the transverse septa. So and some of them are having the longitudinal septa as well. So longitudinal septa are few, but the transverse septa are near about around 10 to 11. So this is the you can differentiate the early blight conidia late with the late blight conidia, which is caused by Phytophthora infestans. So this is the distinguishing feature of the pathogen, that is Alternaria and Phytophthora. So you can see this is the mycelium. So this is the mycelium, which is a septate. As I said, they are having the septa. So when we culture this Alternaria, there are different we can say different colors of the pathogen under, and when we isolate under lab conditions. So we can see the disease cycle of early blight of potato and tomato. So as I think yes, the infected lesions on potato, these are the infected lesions on potato tuber or tomato. So these are the symptoms of early blight in potato and tomato. So the conidia or mycelium get overwinters in the infected plant debris on these tubers or on the fruits. These conidia and they are overwintering, and when there is a favorable condition, so these conidia directly get germinated, and they get they get to form the germ tube, and they penetrate. So and this is called as a direct penetration, and they invades the host tissues, and then it is again infection that lesions will appear on the leaf, stem, and fruit. So this then these new conidia are produced on the infected tissues, and these from this the conidia get detached, and again it forms the germination, and they get penetrated into the tissues, and they get causing the infection in the fruits. So this how that's how the cycle of the Conidia or Alternaria solanae gets repeated. For management of early blight of potato or tomato, so we need to follow the cultural practices, that is removal of the diseased crop debris. 3 to 4 years of crop rotation is required to reduce the pathogen inoculum, or to reduce the or to destroy the pathogen inoculum. So early blight is more important in autumn and winter season. Therefore, adjustment in the planting time is very much required. So to get escape from the disease. And resistant varieties, the Anthracnose is the resistant variety of the tomato. So this is the recently developed Anthracnose is the recently developed variety from IHR Bangalore, and which is having in addition to the early blight of potato or tomato, so it is having the resistance against the virus, that is tomato leaf curl virus, as well as the tomato wilt. So this is the triple resistant variety of the tomato, that is Anthracnose. So for the management of the Alternaria, we need to follow the chemical sprays. Now, mancozeb is effective against mancozeb, or Captain is the effective for the chemical spray. So other fungicide, that is copper oxychloride, chlorothalonil, tebuconazole, are equally effective for the control of the fungal pathogen. So Tomcast is one of the forecast system which is followed for based on the forecasting system. Because in the forecasting system we come to know that when there will be a drizzle, when there will be a cloudy weather, when there will be a change in the climatic conditions. So based on this, we can devise the strategies for the control of the different early blight of this potato and tomato. So these are the forecast system, that is Tomcast is used for these assessing the parameters related to the environment. So in addition to these biological control, like Trichoderma and Pseudomonas species are encouraged to reduce the inoculum load in the field condition. So another important disease of potato, which is black scurf, which is caused by Risottonia solanae. So this is one of the common disease of potato and has a worldwide occurrence. The disease has also been known as a stem canker, aerial tuber, top rosette, web blight, fungal infection, mainly reduces the quality of the tubers and affects the market value, because this pathogen causing scurfing, or the black colored mass on the potato tubers. So that's why the disease name is a black scurf. So after appearing this scurf, we don't get the market value of this crop. So this disease is causing heavy losses to the growers. So initially, this pathogen attack on the young growing tips and sprouts of the plants, and they cause the pre-emergence death of the plants. But plant shows the stunting stem canker on the potato tubers. That's why we are calling it as a stem canker disease. So green aerial tubers appear on the branches. It is the symptoms in affected potato plants by the black scurf. So green aerial tubers are formed. So that's why we are calling it as aerial canker. So dry rot symptoms are prominent when the pathogen enters the tuber from the wounds. So in after get entry in from the wounds, the pathogen cause the dry rot symptoms in the tubers. That's why we are calling it as a dry rot disease of potato. So you can see these symptoms on potato as well as on the stem of potato plants. Okay, the pathogen of black scurf in potato is a Risottonia solanae, is a soil-borne pathogen, and it is affected by the soil conditions, which are different soil conditions like temperature, which is 16 to 23 degrees Celsius is required for its development, and the temperature of about 25 degrees Celsius, which get affected its germination and development. So these are the spores of the these are the spores of the Risottonia. You can see in the picture. This one is the mycelium of the Risottonia, which is again mycelium, branched mycelium. So these are this is the pathogen under in petri plates, so isolated in the petri plates. So these are the different protein structures you can see in the plates. These are the Sclerosia formed by the Risottonia solani. So this one is the greenish mycelium. So there is a fungal diversity. So there will be different colors and morphology of these fungus, because it's a highly diverse fungal pathogen. So this can be different types of Risottonia solani. So now you can see the disease cycle of the Risottonia solani. So the pathogen overwinters as a Sclerosia, nothing but the fruiting bodies of the fungus and mycelia on the infected tubers in the plant residue or on the plant debris in the soil. So the infected tubers get during the spring season. These infected tubers, the mycelia and Sclerosia on the infected tubers get starts germination and start on the developing sprouts. Initially on the developing sprouts, root and stolon primordia, often the nipping of the growing tips of the potato. So and then it causes the infection. And during summer, the roots and stolons may be attacked anytime during the growing season. And in late season, damage may result in the formation of the aerial tuberals. As we know that symptoms of the this yeah, there will be a aerial greenish aerial tubers will be there, as like potato. The smaller, but these tubers will be very small, but it will be green in color. So that's why the name is called as a aerial tubers of the disease. So and that's how the cycle gets repeated. So management of the black scurf of potato, we need to follow the cultural practices, use of green manuring, crop rotation, shallow planting, because if we allow the field to have more moisture, so the pathogen will get perpetuated and developed. And therefore the field should be a good drained field, which helps in the disease reduction. The field can be amended with the different neem cakes, different organics, that is neem seed cake. So approximately 25 quintals per hectare, which will ultimately help in reduction of the pathogen in phylum and the field condition. The chemicals like organomolecular chemicals are required before planting or storing of the infected seed. That is application of dipping, that is 1.75% sulfuric acid for 20 minutes is effective for the control of the sclerotial development. Another important disease of potato, that is water potato, which is caused by Syncytium endobioticum. So this is again zygomycetes. This disease was first reported in 1895 in Hungary, and very serious in Europe, Russia, Peru, Canada, and United States. So in India, the disease was reported from Darjeeling by Ganguly and Paul in 1953. So due to the strict domestic quarantine, it is restricted to Darjeeling and nearby area only. So now it is not reported in other parts of the country. So it is restricted or having due to the domestic the potatoes from the Darjeelings are not transferred from other places of the country. That's why the disease is not spread in other states wherever the potato is grown. So the water potato is also caused in the tomato. So these are the symptoms of water potato. So you can see in the picture that it is a very serious disease caused by soil-borne fungus, Syncytium endobioticum. It is an obligate parasite. Obligate parasite means it requires the living host to complete its life cycle. That's why we are calling it as obligate parasite, which does not produce any mycelium, but an abundance of dissemination sporangia, which are responsible for the tumor formation on underground potato organs. So this one is the requires the living host for the completion of the life cycle, because This does not depend on the alternate or any collateral host or dead plants, we can say, because this fungus is having the enormous sporangia, and these sporangia get transferred from one to another places, and they get affecting the crop plants. So management of water potato, we need to regulatory measures. We need to follow the regulatory measures because it is highly it is very difficult to eradicate once it is uh, infected or once it is introduced in any new soil or new places. So that's why we need to follow the regulatory measures, because only the adjoining area is contaminated with the disease. So we need to follow the strict regulatory measures, so that it cannot be transmitted from this place to the another different parts of the states. So cultural practices, we need to follow at least 10 years of crop rotation with the non-host crops. And steam sterilization of soil is very effective, but it is not a feasible approach because it is a very expensive strategy to manage the disease. So resistance, that yes, resistant varieties are there. So Kufri Sherpa, Kufri Jyoti, Kufri Jivan, Kufri Mutun are the resistant varieties both for the late blight as well as the wart disease of the potato. So these are the other important diseases of potato which I have enlisted. So that is powdery scab, Spongospora subterranean, powdery mildew, Erysipelas coriarium, stem blight, Sclerotinia sclerotiorum, Cercospora leaf spot, Cercospora concourse, tuber rot, Fusarium solani, silver scurf, that is Helminthosporium solani. As like black scurf, silver scurf is another disease which is caused by Helminthosporium solani. Anthracnose, as we yesterday we talked about the anthracnose. So anthracnose of the grapes. So this caused by Polytrichum cocodes, whereas the anthracnose of the grapes is caused by Polytrichum cleosporites. So dry rot, that is Fusarium solani. So wilt, again the Fusarium solani. Charcoal rot is again one of the important disease which is caused by Macropomena phaseolina. So these are the other diseases of the potato. So now we have covered potato and tomato because same pathogen are causing the diseases in potato as well as in tomato. So like early blight, late blight, and even wart. So anthracnose is one of the important disease. As yesterday we have seen the details about anthracnose, only symptoms, the because the rest of the life cycle, disease cycle, and the pathogen will be the some pathogen structure will be different, but the disease cycle will be the more or less same, and the different strategies of management will be slightly different. So just we can see the symptoms of the anthracnose of chili. You can see in the picture that these are the some concentric rings are appeared on the fruits of chili. Similarly, these are the concentric rings occurred on the beans. So you can see in the picture. So these concentric rings or lesions, they join together and they cause complete damage to the beans. So these are the brinjals which are affected by the anthracnose. You can see in this brinjal also there are the concentric rings of these which is caused by, this is the characteristics of the anthracnose, and these are the concentric rings are caused by two pathogens, that is anthracnose, that is Polytrichum, and by the Alternaria. So these are the typical symptoms of the two diseases. So this is a list of fungal diseases which are reported to cause infection or different. These are the different diseases caused by different fungus. That is anthracnose. Again, it is caused by Coleotrichum gliosporites, Coleotrichum capsicum, Glomerella cingulata, which is a teleomorph, or we can say it is a perfect stage of the Coleotrichum. Polytrichum cocodes, perfect stage we can see because this perfect stage is the stage of the pathogen where it can produce the sexual spores. So the Cercospora or frog eye leaf spot is occurring in chili, which is caused by Cercospora capsicum. Charcoal rot, Macropomena phaseolina, as like in potato, in it is affecting also chili also. So wet rot, damping off, damping off and root rot. Damping off and root rot are the diseases which are causing large number of vegetables, which is caused by Risottonia solani, Pythium, Fusarium, and Phytophthora. So these are the important pathogens which are causing the damping off. Based on the different symptoms, they are named. If it is causing the rotting of the roots, that's why it is called as a root rot. So damping off because it is causing the disease at the early stage of the crop growth. So that's why the pathogen and the seedlings get damaged, seedlings get collapsed, and that's why we are calling it as a damping off. Downy mildew, Peronospora tabacina, Fusarium stem rot, Fusarium wilt. Yesterday we have seen that in first lecture we have seen that Fusarium wilt of sugarcane. So in that I have explained about the pathogen. So gray leaf spot stem phylum, which is caused by gray mold, Botrytis cinerea. So Phytophthora blight, Phytophthora capsicum, powdery mildew, Oidiopsis sicula, southern blight, Sclerosium rodsai. These are the teleomorphs of these pathogens. Verticillium wilt, Verticillium, white mold, Sclerotinia, Sclerosiorum. So these are the different diseases of the chili. Now we will see the diseases of onion and garlics. So one of the important vegetables in India, one of the important disease of the onion and garlic is the purple blotch, which is caused by Alternaria. Again, but the species is different, Alternaria pori. So the symptoms that initially small elliptical lesions or spots that often turn purplish to brown, which are surrounded by chlorotic margin. These are the spots which are caused by the purple blotch pathogen, that is Alternaria pori. If the spots enlarge, the chlorotic margin extend above and below the actual lesion. The lesions usually girdle the leaves and causing them to fall over. The lesions may also start at the tips of older leaves. So for the management or control of the pathogen, that Alternaria, application of different chemicals are required. That is mancozeb at the rate of 0.25%, tricyclazole 0.1%, hexaconazole 0.1%, propiconazole 0.1% at 10 to 15 days interval from 30 days after transplanting, or as soon as the disease it appeared in the field condition. We need to follow these chemical sprays avoid the damage due to purple blotch. So second important disease is the stem phylum blight, which is caused by again stem phylum vesicarium. Though if you can see the pictures or symptoms of the stem phylum blight. So initially small yellow to orange flakes or sticks develop in the middle of the leaf, which soon develop into the elongated spindle shaped to ovate elongated diffused spots, which are surrounded by pinkish margin. So you can see in the picture that there is a pinkish margin, and these are the spots of the stem phylum blight. These spots progress from the tip and to the base, and the foliage and extending the patches and the blighting the leaves, and gradually the entire foliages get damaged. So for the management, again the similar kind of chemicals work for the management or control of the stem phylum blight. So third important disease is anthracnose. So Trichome gliosporides, the same pathogen which is causing anthracnose in the grapes. So here the symptoms are similar, like it is causing the concentric rings on the leaves, the characteristics of curling, twisting, and chlorosis. These are the additional symptoms of the leaves. Abnormal elongation of the neck. So the stem get damaged, neck get damaged in this. Initially, the pale yellow water-soaked oval sunken lesions are appeared on the leaf blades. Numerous black-colored, slightly raised structures are produced in the central portion, which may be arranged in a concentric rings. The affected leaves shrivel, droop down, and finally wither. For the control of these anthracnose, the application of benamine 0.2% and 0.25%. Then planting onion on the raised beds is recommended, and we need to avoid the waterlogging conditions. Another important disease that, as I said, damping of root rot, soft rot, these of vegetables which are caused by Pythium species. So these diseases are occurring in different vegetable crops. So combinedly, we can study the nature of the disease and the causal pathogen, which is epithelium. The species may be different, but the management strategies and the disease cycle will be more or less same. So initially the young seedlings are affected before the emergence at any point on the plant. So infection spread rapidly and invaded cells collapses and seedlings is overrun by the fungus and dies. That's why we are calling it as a damping off or pre-emergence. Damping off means before the emergence of the seedling. So this infection is occurring inside the within below the ground level. Okay, so that's why pre-emergence or damping off symptoms are caused. So already the emerged seedlings are attacked. If there are attacking in the already emerged seedlings or established seedlings, the the pathogen attacking the roots and stems, which is below the soil line. The invaded area becomes water soaked lesions, discolored, and it get collapsed. The entire plant or seedlings get collapsed. The basal part of the seedling becomes softer, thinner, and as compared to the healthy plants. So it cannot bear the weight of the seedling, so that it get damaged and it get collapsed. I will show the symptoms. So these are the symptoms of damping of root rot and soft rot in different vegetables. You can see this is the foot rot symptom in the cucumber. So this is damping of symptoms in the watermelon seedling. Similarly, damping of symptoms in the cucumber. The foot rot symptoms in the pumpkin, foot rot in watermelon. So this is a foot rot in tomato. So these are the different type of symptoms caused by the pythium fungus. Now we can see the life cycle of the pythium. So these are the infected plants or seedlings. So bearing the sporangiophores and sporangia. The sporangia get detached from the infected plants or infected seedlings, and they form the vesicles, and these vesicles indirectly. So because there are again the two ways of sporangium infection. So when there are the optimal conditions or environmental conditions, these sporangium directly form the germ tube and they cause the infection to the host. Whereas, so if there are environmental conditions are not suitable for the germination of the sporangium, it forms the vesicles. Vesicles are nothing but the protein, the structure which is containing the zoospores. So they get mature here, and whenever there are optimal conditions, there are moisture conditions available in the field, so they get burst and the zoospores are released. These are the zoospores. Zoospores are the motile spores. They are having the flagella. This one is the flagella, that is biflagellate zoospores are there. So these zoospores get in the after the getting the moisture, they get translocated from one place to another, and when they get the suitable surface for their attachment, the spores get the flagella get detached, they get encysted, and these zoospores form the germ tube, and they get germinate, and they cause the infection to the host. So for this is the asexual life cycle of the fungus, whereas in case of the sexual life cycle of the fungus, there are the two mating population, that is male and female parents. So the Anthridium is the male parent and Oogonium is the female parent. So this bulged or circular shape is the Oogonium, and this one is the Anthridium. So they follow the meiosis. They are followed by karyogamy, and they form the oospores. So the oospores are nothing but the resting spores, and these oospores last maybe more than a year or many years under uh, when there are not suitable conditions or environmental conditions for their growth. They get overwinters in the infected plant parts. They remain in a dormant situation for a long time, and when there is a conditions which are favorable for their germination, they forms the again there are the different strategies. They directly, if the conditions gets immediately gets suitable for their germination, they forms the germ tube, and they causing the infection to the host. So and they form they follow the secondary pathway, that is indirect infection. So they form the juice pores, they get incysted, and it causes the infection to the host plants. So this is the life cycle of the pythium. So this is common for all the pythium species, which are causing different diseases: soft rot, foot rot, damping off in different vegetables and fruits. So again, we need to follow the different management practices, starting from the cultural uh, to the resistance cultivation of the resistant varieties, because this is uh, uh, zygomycosities. So if we manage the field, well drained field, so we can reduce the level of inoculum, primary inoculum, so that it cannot multiply and it cannot cause the. So in case of the nursery conditions, the pathogen is become very severe. So in nursery, this is the very problematic pathogen to manage. That's why the seedlings are properly maintained under the well-drained conditions, and the seedlings should not be under the submerged conditions. So and after transplanting the seedlings to the main field, the seedlings are treated to the different fungicide like bavistine or Captain or Thyram. So these seedlings are treated with different fungicides, systemic fungicides, so that there won't be transfer of propagule or pathogen from nursery to the field conditions. So these steps has to be steps has to be followed. So and so that we can manage the rotting or foot rotting or damping off or soft rot of vegetables. Okay. So these are this is all about the fungal diseases of vegetables. So we can stop here for 5 to 10 minutes and we'll rejoin again, and or